This chalk talk will cover frontotemporal lobar degenerations, or FTLDs. This is a little bit more complicated than some of the other talks on these entities, but I've tried to streamline it and keep it as simple as possible. So let's have at it. Frontotemporal lobar degenerations are a heterogeneous group of degenerative dementing diseases that share several clinical features and are characterized by selective frontal and temporal lobe atrophy, as the name suggests. These account for about 12 to 20% of cases of dementia, so it's an important category of dementing diseases. The clinical presentation is variable between cases, but are clinically distinguished from Alzheimer's disease by alterations in personality and behavior, or language in the form of aphasias, that precede the onset of memory loss. There are several clinical variants in which either behavioral changes or aphasias dominate the clinical picture. Global dementia occurs with progressive disease in all forms. The clinical pathological syndrome for these related disorders is frontotemporal dementia, or FTD. Now, historically, diseases falling into this category were grouped together as Pick's disease. Modern insights from immunohistochemical and molecular studies have revealed several distinct types of frontotemporal degeneration, and the term Pick's disease is now restricted to cases of frontotemporal degeneration associated with widespread Pick bodies. So we'll talk about that in a little bit. As with many neurodegenerative diseases, FTLD is also associated with cellular inclusions of specific proteins. The two most common patterns are those with tau-containing inclusions, these are called FTLD tau, and those with TDP43 containing inclusions, conveniently called FTLD TDP. And these account for about 95% of FTLDs. Within each of these groups there are heritable forms as well as sporadic cases. There is no fixed relationship between the clinical subtypes of FTLD and the types of neuronal inclusions. Most subtypes of FTLD tau have decreased brain weights with varying degrees of frontotemporal atrophy. Mesial temporal lobe structures are most severely involved, usually also the putamen, caudate, globus pallidus, hypothalamus, and often the orbital and cingulate gyri. So a little bit different neuroanatomical distribution compared to Alzheimer's disease. Generally, FTLD tau shows varying degrees of neuronal loss and astrocytosis in the frontal and temporal lobes, as well as in the hippocampus, amygdala, and subiculum. Typically, superficial spongy change in the affected neocortex is evident, usually in the second neocortical layer. Tau deposits are found in neurons, astrocytes, and oligodendrocytes. Their morphology, cellular and regional location, and density vary with the specific mutation involved. They may be found in the cerebral cortex, white matter, cerebellum, and brain stem. So this image shows the localization of this vacuolation or spongy change in the cortical layer, neocortical layer 2, and there's relative preservation of the deeper layers of the cortex, and this is characteristic for frontotemporal dementia tau. As mentioned, you can categorize FTLD with tau immunoreactivity. To the left, neocortical neuronal and neuritic tau immunoreactivity is demonstrated. The middle image shows neuronal and neuritic immunoreactivity in the dentate gyrus. And finally, on the right, there's a glial tangle and neuronal cytoplasm staining with tau in the temporal cortex. Pick disease is a subset of FTLD tau in which the atrophy is typically extremely severe in the frontal and temporal lobes, often referred to as knife edge or walnut-like. The essential finding in Pick disease is the Pick body. So whole brain images here of the lateral and ventral aspects show diffuse severe atrophy affecting the frontal and temporal lobes, as well as in the parietal lobe. You can see this demonstrated on the ventral view as well. Note the severe bilateral atrophy, especially in the temporal and frontal lobes here. This is reflected histologically in this image with neuronal loss and gliosis affecting all cortical layers all the way down to the underlying subcortical white matter. To repeat, the essential finding in PIC disease is the PIC body. This spherical cytoplasmic inclusion is found in neurons in the frontal and temporal cortices and also in the mesial temporal lobe structures, especially in the dentate granular layer of the hippocampus. These are gyrophilic or silver-loving and immunoreactive for proteins tau and ubiquitin. PIC cells are another feature, which are essentially surviving eosinophilic swollen neurons. The upper image is a silver-stained section of the dentate granular cell layer of the hippocampus, showing numerous PIC bodies in the cytoplasm of the dentate neurons. So if you look closely, the darker staining round objects here are the PIC bodies, and the sort of lighter staining brown are the nuclei. This is more evident on the lower image, which is immunostaining of the temporal lobe cortex, showing positive staining for intracytoplasmic spherical PIC bodies, which displace the blue stained nuclei. This is an immunohistochemical stain for tau protein, again demonstrating PIC bodies within the neuronal cytoplasm. These slightly less intense staining are the neuronal nuclei. And finally, these images demonstrate PIC cells. In the upper image, you can see multiple ballooned chromatolytic-like neurons in the amygdala of a patient with PIC disease. The lower image shows the same type of cell, only this time with immunostaining to phosphorylated neurofilament. Next, I'll draw some contrasts with FTLD-TDP. The brains of patients with FTLD-TDP usually exhibit decreased brain weight and cortical atrophy, which is, again, most severe in the frontotemporal lobes. Atrophy may also be very severe in the mesial temporal lobe structures, and then compensatory ventricular dilation occurs. Atrophy of the basal ganglia may also be present, and the atrophy may be asymmetrical. Microscopically, there is variable neuronal loss and astrocytosis or gliosis in the frontotemporal lobes. Laminar spongiotic change is seen in a little bit wider band than in FTLD tau, involving neocortical layers 1 to 3. You can also see the ballooned neurons, as well as hippocampal sclerosis, which is frequently evident. The key, however, are the immunohistochemical features identifying TDP43 protein. TDP43 is normally localized to nuclei. However, in FTLD-TDP, it's relocalized to the cytoplasm, where the diagnostic inclusions are formed. TDP43 immunoreactive inclusions are present in the hippocampus, neocortex, striatum, globus pallidus, thalamus, substantia nigra, the white matter U fibers, and spinal cord. So they're widely distributed. There's a variety of different TDP43 positive inclusions that are described. They can be intranuclear in neurons or cytoplasmic in neurons. They can be associated with dystrophic neurites. They can also neurons can also show diffuse cytoplasmic granular staining, and you can also see cytoplasmic inclusions in glial cells as well. So the image on the left shows immunostaining of dentate gyrus neurons. So these are intranuclear inclusions from the hippocampus with TDP43 antibody. To the right, you can see TDP43 positive cytoplasmic dot-like inclusions in the absence of intranuclear inclusions. Alright, so let's summarize. This is my game plan for studying FTLDs. These are a heterogeneous group of disorders associated with focal degeneration of the frontal and or temporal lobes. They're distinguished from Alzheimer's disease by alterations in personality and behavior, or alterations in language in the form of aphasias that precede the onset of memory loss. Clinically, these are referred to as frontotemporal dementia. Grossly, all forms are associated with decreased brain weight and cortical atrophy, which is most severe in the frontotemporal lobes. Pick disease is an exaggeration of this, in which there's extremely severe atrophy in the frontal and temporal lobes, often referred to as knife-edge atrophy. In general, microscopically, there are varying degrees of neuronal loss and astrocytosis in the frontotemporal lobes, typically involving the superficial cortical layers. However, in Pick disease, the neuronal loss is more extensive, affecting all cortical layers, and is associated with marked gliosis and the presence of hallmark Pick bodies. And finally, we can characterize FTLDs biochemically according to their immunoreactivity for tau deposits located in neurons, astrocytes, and oligodendrocytes, the presence of Pick bodies, which are also immunoreactive for tau, as well as ubiquitin. In contrast, FTLD TDP is associated with TDP43 immunoreactive inclusions, which can be localized to nuclei or cytoplasm. So that's it for FTLDs. It's a complicated, diverse subject, but hopefully I've made it more straightforward for you to learn. Thanks.